Today, I'm going to talk about who we are and who we will become, and how the field of developmental cognitive neuroscience will start to answer these questions. And I usually like to start with a video of a newborn, right? So as you can see, this newborn is sleeping. Newborns sleep a whole lot, right? And you can see that her eyes are moving around. She's probably experiencing REM sleep, maybe dreaming. The question is, what is she dreaming about, right? She was just born. What is she thinking? What does she know? And who is this person going to become? And these are questions that humans have asked themselves for centuries, right? What makes me me? And is it what I was born with, or is it the experiences that really shape the person that I am today? So it's the classic nature versus nurture debate. So to formalize these questions, right, how much of the human mind is determined at birth, and how do we gain new skills that are uniquely human? Is there a limit to our potential based on the stuff in our brain that we're born with? But what if I were to tell you that science is now at the point where we can start to really answer some of these questions? We're able to do experiments that we could only dream about doing 40 to 50 years ago. So let's see if we can answer some of these questions with brain imaging. And so here's an MRI scanner. I don't know how many of you have been in one of these or had the opportunity to see one, but this is what it looks like. You climb in here and you go into the scan bore, and we can get these types of images from it, right? So here's a baby scan. You can see brain anatomy, like where stuff is in the brain. You can get brain function images, so what each brain region is doing when you're performing a mental task inside of the scanner. And you can get these really cool connectivity images of what brain regions are connected to one another, the strength that they're connected to each other, and you can visualize all of these pathways in 3 dimensions, which is amazing. So using all of these neuroimaging methods, here's what we know about the adult brain. Right, so here is an adult's brain, and I inflated all of the crevices such that we're looking at the entire surface of the brain unfolded. And we turn it around such that we're looking at the underbelly. So the eye should be here, the back of the head is here, and we're looking at it from this way. So we're looking right here in temporal cortex. And we know that the adult brain has a very rich sort of organization, and there are specific brain areas that do very specific mental tasks. So this is one of my favorite brain areas. These brain regions help us perceive faces. So whenever you're looking at a face, whenever you're looking at my face, right, these are the brain areas that are engaged and activated. And if you lose these brain areas, like with stroke or any damage, you're unable to perceive faces, even familiar ones. And that's a condition known as prosopagnosia. Other visual areas are responsible for how we perceive everyday objects like chairs, clocks, whatever, right? All the stuff around us. Other brain areas help us perceive scenes. So when we're navigating through the world, when we come into a new place, these brain regions are active. It's like a GPS system that's built right into our brain. And here's a brain region that I'll talk about in some depth today. It's called the VWFA, or the visual word form area. And this brain area is responsible for helping us read. So it processes orthography. Other brain areas help us understand what it is that we're reading. So these are the regions in yellow, and these are the language areas, right? So they help us know what people are saying, they let us talk, and they let us communicate with one another. Apart from these rather domain-specific brain areas, we also have brain regions that are more general. And these regions help us attend to things in the environment, and they help us filter out irrelevant things. So these regions are more general purpose. Whenever you're focusing hard on something, when you're trying really, really hard, no matter what it is, right? It could be math, it could be trying to read something, understand something, listen to someone. These regions are activated if you're focusing hard. All right, so does this organization exist at birth, right? So all of these brain areas are present in every person that we scan, and they're in approximately the same place. So, it's kind of weird, especially that some of these brain areas show up in the same place. So we're wondering what drives this functional organization of the brain. And does this organization already exist at birth? Are we born with it? And we're now at the point where we can peek inside a baby's brain and we can compare this brain to other brains, and we can use computational modeling and artificial intelligence to try to predict things like future outcome and behavior in each individual baby. So imagine being able to know what your child is thinking about and how you might be able to help them reach their highest potential. It would truly be revolutionary. And so that's what we're working on, but it takes a lot to scan kids, right? It's hard and not every lab can do it, but the questions are just too interesting to not try it. In my lab, when I got here, we spent quite a bit of time trying to figure out the best way to collect high quality data, and we've converged on a set of protocols that we now implement. So for newborns and infants, we wait for them to fall asleep and then we scan them. We swaddle them, mom feeds them, and we wait for them to fall asleep. And once they're asleep, we move them to the scanner and an experiment. This is me here. An experiment is with the baby throughout the whole scan. For children, for a bit older children, we do as much as we can to prep them before they come into the MRI. We send kids a video to watch before they come in, and we've modified our video slightly for the current pandemic state. So here it is.
Welcome to Z-Lab, the Saygin Developmental Cognitive Neuroscience Lab. In our lab, we study how kids like you learn and remember things, how plasticity happens in the brain as we learn new things, how it makes us into different people, and how it is able to recover from any injury that might happen to it. We learn about these things when kids like you participate in studies like you're doing today. We're going to show you what to expect with an MRI scan in our lab. So first, after picking you up in the lobby, we'll take you upstairs to our lab. And this is our lab here. This is where we'll learn the games and fill out some papers. While in the lab, during the pandemic at least, we'll take your temperature like this. In the MRI scanner, you'll have to wear earplugs because it's really loud. We'll have you practice in the lab, just like Athena here. She's one of the researchers here at OSU, and she might even be the one helping you today. After we learn the games, we'll go from our lab to an elevator that will take us downstairs to the MRI scanner. But before we go in, we'll make sure you go to the bathroom one more time just to be sure. And then you'll meet Darren. He runs the MRI scanner and ensures your safety. To make sure you're safe to go into the scanner, he'll use a special wand to make sure you don't have any metal on you. You can see him checking our scan dog here, and upon ensuring there's no metal, he'll allow you to go into the scanner. You'll lay on the table like our scan dog here. We'll help you put your earplugs in so it's not so loud, and Darren will put a mask over your head so that we can get good pictures of your brain. And then we'll give you the buttons to push for the games. Then Darren will move you into the scanner like he does with the dog here. You won't be alone. Someone will be with you by your feet the whole time. And after the scan is over and we have all the pictures we need, Darren will move you out of the scanner one more time and you're done. You can go collect your toys and snacks because you helped us to do some science.
So yeah, kids get this before they come in, and after the scan, we give them compensation, but we also give them snacks and small toys, and they get them, and they know that they're getting them after they complete each short scan. So it's motivational for them. So in my lab, we try to answer the overarching question of how does the architecture of the mind arise? And we study children, neonates, and developmental disorders. And I'm going to give an example from each of these three branches of study in my lab, and I'll use reading as the overarching unifying theme. All right, so as we know, there's this region of the brain that's critically involved in reading, right? It's called the visual word form area, or the reading area. And what makes reading such a great place to start is that this region that's involved in reading doesn't exist before reading, right? It exists as in the brain cells exist, but the cells in this region don't know the difference between words and other visual stimuli. So we say that this region doesn't exist in pre-literate kids or adults. But there may already be the right type of connections that are set up in this region, right? Maybe this region is just waiting for the right type of input to become an orthographic reading area. So to answer this, we scanned kids before they knew how to read at age 5, and we got a connectivity scan on them. And then we brought the same kids back at age 7 after they learned how to read, and we got a functional MRI scan on them. And that was in order to delineate where the VWFA was in each child's brain. And we wanted to test whether early connectivity at age 5 could predict the exact location of the VWFA in that same child at age 7. So before I show you the results, we're going to inflate the brain again. And so remember that we're scanning kids before they knew how to read. And then we're scanning them again after they learned how to read. And we're going to see whether we can predict the location of the VWFA in each child. And the region varies in its exact spatial location in every child. So we wanted to know how precise could we be and how well we could predict its exact location in each child individually. So here is a child's actual fMRI data when they're looking at words and in the scanner, and word selective cortex should be here, circled in magenta. And we know this because we scanned them at age 7, and this is where word selective cortex is going to show up. And this is what was predicted using the same child's connectivity data alone at age 5. And what we can see immediately is that a lot of this word selective cluster can be very well predicted from the same child's brain scan at an earlier age. So we can use a brain scan at an early age to predict where in the brain of each child a reading area will show up. It turns out that this brain region is already connected to a set of other brain regions like language areas that set it up to become selective to written words. And so we're putting together a wiring diagram of the circuitry to better understand how it works and how it develops. So early connectivity seems to be earmarking the location of the VWFA, a region that's entirely experience dependent. Right? But this evidence wasn't entirely satisfactory. So next we explored whether this connectivity pattern was already present at birth. So when you're asleep, or even when you're just spacing out, as you may have done a few times already, your brain is still active and busy, right? So it turns out that regions that are involved in similar mental functions fluctuate together. So they're going up and down at the same rate. So what this means is that regions are connected to each other functionally. They're involved in similar mental functions. And it turns out that when a baby is sleeping, this is what's going on in that baby's brain. So this is a neonate brain scan. And so what we did was we compared functional connectivity in neonates to functional connectivity in adults. And we wanted to know whether this network, the circuitry, was innate. And we looked at the connectivity of these language regions. And what we found is that neonates, just like adults, have parts of visual cortex that are very well connected with language areas. And it so happens that that the language network connects more to what's later going to become this reading area, more so than all of these other areas. So it's not connected to face areas, it's not connected to scene areas or object areas. It's connected to what's going to become the VWFA. So a region that will later develop orthographic selectivity is already connected with language cortex even at birth. So pre-existing connectivity may be a way for a visual area to become language sensitive. Now I'm going to turn to the last example of today's talk, where I ask whether connectivity can predict later behavior and later clinical outcome. And specifically, we looked at whether we could predict which child would show reading difficulty or develop dyslexia. We know, for example, that there appears to be a circuitry inside the brain that's different in older kids who have dyslexia. So certain tracks, the ones that I've highlighted in color here, they tend to be different in older children and adults with dyslexia. One of the more important ones is this tract in yellow. It's the arcuate fasciculus, and it connects the frontal and the temporal language areas. So we wanted to know, are these differences the cause or the consequence of dyslexia? So are any of these tracks different in pre-literate kids who may later go on to develop dyslexia? If we do see evidence of this, it might mean that these differences are causal, and at the very least, they can be used as biomarkers of dyslexia. And what we found is that the volume of a particular white matter tract, the left arcuate, was correlated with a pre-reading measure of dyslexia, namely phonemic blending. So rendering a few subjects' arcuate fasciculi in 4 children, you can literally see this difference, right? So a child with lower phonemic blending ability has a smaller arcuate than a child with superior blending ability, and these are all normalized for head size. So the differences in this reading circuitry that we see in older children seems to exist even in pre-readers. And so we can make predictions about who might go on to develop dyslexia using a child's MRI data even before they know how to read. And this might help introduce early interventions and can potentially prevent issues with years of struggling to read. So I showed that early connectivity can predict the reading area location in each individual child. And that's pretty cool, right? Because what that means is that we can use non-invasive neuroimaging methods to predict what the brain will look like years from now. And that would be groundbreaking, right? We also went even earlier and we found that neonates already have this rich circuitry set up, which suggests that innate mechanisms may determine what the brain might do years later. We then showed that not only can we predict the organization of the brain as it develops, but we can also predict learning and behavior using early brain scans. And this can potentially help issues with reading even before a child starts to have trouble. But we're just getting started, right? So we're continuing to do longitudinal studies on children starting as early as possible. And we're following up with them as they get older to see how well we can predict developmental milestones like walking and talking, as well as school-age skills in each child using early brain data. And I talked about reading today, but we're also looking at other skills like math, attention and cognitive control, memory, emotional processing, and social processing. So we want to know, how well can we predict these milestones from early brain data? We also have some work that I didn't have a chance to talk about today that looks at how the brain changes across time, right? So how does experience and maturation change the brain, and what types of experiences lead to what types of changes in brain and behavior? But I'm going to take a step back and quickly talk about why this work is so important, right? So why should the world care about developmental cognitive neuroscience? So studying how the human brain and mind develops and making individual subject predictions has both scientific and practical importance. For basic science, if we can use cutting edge methods in cognitive neuroscience, and if we can combine it with cutting edge methods in artificial intelligence, and we can try to predict developmental milestones, we're going to establish where we are as a field in understanding how the brain gives rise to behavior, right? So we can't say that we truly understand how the brain works without being able to accurately make such predictions. But on the flip side, even if we are somewhat inaccurate at making these predictions, we will know where we are as a field and what we need to improve on, right? So we can actually test these things non-invasively in humans. This work provides a way to test the nature versus nurture debate, as well as understanding what about our brains make us unique, how we learn, and why childhood is so important, as well as what would happen to the developing brain if there were certain changes in experience. This research also has educational applications, right? Can you imagine if a brain scan could predict reading ability, math ability, and so on? So this could be a game changer for individualized and adaptive education. And the technology that's built from these findings could potentially shift existing educational paradigms. This work also has broad clinical applications, right? So if we're able to predict disorders even before they are apparent or even before the skill can even be measured, like in the case of predicting reading ability in pre-readers, we can try to develop interventions even before a child starts to have trouble. Interventions, remediation, and targeted practice will greatly improve the quality of life of individuals, and it might even prevent the development of certain disorders. All right, so this work is important. What is the future of this field? And I think that the future of this field is in merging big data with artificial intelligence, right? So using machine learning methods, much like the stuff that I presented here, to understand how the brain makes the mind and how it can change with experience and development. It's also in developing innovative technologies like MRI head coils and stimulation devices to make groundbreaking discoveries in cognition and development. The future is also an open and inclusive science. So we can't get to these goals alone, right? We need to collaborate and we need to share data, increase diversity, and greatly enhance scientific communication and education for our community. So what's the future of this field? It's OSU. So Charles talked a little bit about this, but I'm going to go from the faculty side and researchers side who's currently doing this type of work, right? So our department and OSU in general is a hotspot for all of this work. We have, as you can see, the state-of-the-art imaging facility right in our building. We have a high-performance supercomputing cluster at our fingertips. So it's the Ohio Supercomputing Cluster. And we're developing these cutting edge technologies and making groundbreaking discoveries with award-winning faculty and researchers. So we have a really rare opportunity to be at the forefront of this new movement. And to help get us there, I'm leading an effort to develop a child study center that aims to put OSU at the bleeding edge of science and technology, and that will attract world-class talent. And I envision the center to be the catalyst for innovation and discovery about what makes us us and how we can use this information to make positive impacts on society. So in sum, cognitive neuroscience is now at the point where we need to be studying the developing human mind and brain longitudinally. And we need to work together to standardize procedures, pool our resources, and we need to take the time to dream up the best science possible such that we can inspire the new generation of scientists, policymakers, educators, and hopefully that we will be able to have practical advice for parents to help their little ones achieve their highest potential and therefore help shape the future of society. All right, so I would like to thank my lab. As you can see, we managed to get a Zoom background in nicely with our with our lab meetings. And I would like to thank my funding resources, and I'd like to thank you all for listening to me and to give me the opportunity to discuss my work with you.